the Nigerian Young Academy, the NYA webinar series 3.1. And without wasting our time, we need to start the program formally. So we have the order of presentation. The webinar will last for the next 90 minutes with the opening address to be taken by NYA president, and that will be followed by introduction of Professor Happi, and thereafter we have his presentation for 25 minutes. That will also be followed by introduction of our own Professor Rosemary Audu, and afterwards we have a presentation for another 25 minutes. Then we'll take questions for the next 30 minutes. And the order of raising hand is important for calling on participants to ask their questions. Lastly, we'll be having a vote of thanks by the webinar committee chairman. Thank you. So without wasting time, I would like to call on the president of the Nigerian Young Academy, and that is no other person than Dr. Awal Ibrahim. President, you can take over. Thank you.
Yeah. Okay. Maybe you need to stop sharing. Thank you so much, Dr. Abdulaziz Anjorin, for the beautiful presentation and for also agreeing to take over to moderate this webinar. Please, can you stop sharing? Yeah, thank you. Once again, thank you to Dr. Anjorin for agreeing to moderate and for the kind introduction. I also want to thank the panelists, our distinguished professors, Professor Happi and Professor Audu, who are going to talk to us today. Thank you so much. We are indeed very grateful for accepting to participate in this webinar. I also want to use this opportunity to welcome the participants or the attendees for taking your time to be with us this afternoon to hear from experts in the field, especially with respect to COVID-19. The NYA is actually a unified platform of excellent young researchers, and one of the primary focus of the academy is actually to promote science as well as the uptake of science in policy making across the country. And it is because of that we decided to also, because COVID-19 is a very trending issue, we decided to put forward to us, to the general populace, so that we hear from the experts. Of course, talking about the experts, they are well-renowned scientists. Professors Happi and Professor Audu are almost, I would say, like household names as far as COVID-19, and more specifically as far as COVID-19 and also as far as genomics are concerned. And so therefore, I will employ the attendees and all of us to kindly listen to these distinguished speakers and experts in the field so that we learn a lot, and we will also hear from firsthand, we also get firsthand information from these experts. And also permit me to also say that to all of us, especially the attendees, that this is just one of the series of webinars that the NYA intends to bring in the very near future. And we expect to bring experts in the different fields, but within the COVID-19, to present, to give us very scintillating lectures and educate us on the different aspects of the disease. Like I said, most of the webinars are going to be mainly focused on COVID-19 because it's actually part of a grant from the COVID-19 Rapid Grant Fund, and is actually part of it. And we're also very grateful to the donors. Now, with this, I think I will now just allow the first presenter or the moderator to take over. And I assure us that we are going to have a very scintillating lecture, a very interesting one, judging by the competence and experience of the presenters. Once again, thank you so much, and I welcome everyone to this very important webinar, first webinar in 2022. Thank you, sir.
Okay, thank you very much, President NYA. Without wasting time, I would like to call on Professor Christian Happi. Professor Christian Happi is the Director, Africa Center of Excellence for Genomics of Infectious Diseases, that is ACEGID, at the Redeemers University in Ede, Osun State here in Nigeria, is to talk to us on genomics and translational research in Africa. How close is Africa to local vaccine production? Prof, distinguished teacher of virology, you have 25 minutes and you can start, please. Thank you, sir. Over to you, Prof.
Thank you, and thanks for the opportunity from Nigeria Young Academy to actually for me to give this talk. My talk is going to be divided in two. First, given this topic, that is genomic and translational research in Africa. So I'm going to present some aspect of genomic and translational research in Africa. And in the second part of my talk, I'm going to be focusing on this thematic that is very close to all of us. So how close is Africa, to local vaccine production? So as a way of introduction, I just want to first of all mention the fact that genomics has really become a revolution, and especially with the advent of genome sequencing technologies, and it has revolutionized the way we really approach, healthcare and research. These days, everything about healthcare and research is pretty much driven by genomics. We're talking about personalized therapies and others. So, but then the bottom line is that many of these technological advancements that are driven by genomics, and as well as the economic benefit that comes along with it, are yet to reach Africa. And the reason are just multiple, but the major two drivers for this lack of Africa participation to this genomic revolution is the fact that there's no know-how on the ground, and then there are also no infrastructure. And this has created a huge gap between Africa and then the rest of the world. And then that's the reason why, in order to reduce the gap, we thought about how we can actually get into the genomic revolution and also reduce the divide that is existing between Africa and the world. And how do we want to do that? I think it's about thinking about how you could use what you have in order to actually move faster. And one of those things that we thought that we could actually leverage is the prevalence of all these pathogens that have pandemic or epidemic potential. And if you look at this map, you look at the distribution across this world, SARS-CoV-2 and others, but you can see that the major concentration of those pathogens are in Africa, and which means that it is really something that Africa could leverage in order to reduce the divide and the gap, and also make major contribution to the global community of knowledge, and also position itself as a leader or the driver in infectious disease innovation, and then innovation and that what we did at the time, a few years ago or 6 years ago, is to create this platform called the African Center of Excellence for Genomics of Infectious Disease, built on long-term partnership between some academic institutions and clinical testing centers in West Africa, and then brought in on board international partners. This has really changed a lot, since we started it. But then in order to really do this, we really thought that we should really set up a mission. So we have two major goals in our views. One was educational, which was basically to actually drive start bringing to the fore the genomic agenda and creating an environment where people can actually do, real high quality translational genomic-based research for health. But they also harness this technology and build a critical mass of well-trained African scientists in order to empower them to utilize, genomic tools toward prevention, control, or elimination of infectious disease. In order to do that, we really felt that knowledge, transfer was very important. We created some curricula, genomic curricula. We are the very first two genomic curricula for health in Africa, internationally accredited. So for master's and a PhD program in genomics at Rosemary University, but we also engage communities in prevention effort and in public health, education. So, and this education program was driven by research, as you know, as postgraduate level. And then that research very much is actually about characterizing, the drivers of febrile illness across Africa using microbial metagenomics. And this really helped to drive the agenda to understand the microbial landscape. And it is through understanding the microbial landscape that you could develop the countermeasures. And the countermeasures are diagnostics, therapeutics, and then also vaccines as one of the things that we'll be talking about today. So in order to, and ultimately in bringing this research and then education agenda together, we basically are, meeting one of the critical needs for genomic research and education in Africa. And ultimately the goal for us is actually to build a next generation of African pathogen hunters that will be able to, identify these pathogens, characterize them, and basically develop the countermeasures on them before they get to us. You agree with me that the world is in dire straits today simply because we cannot. I understand the participant cannot see my presentation. Hello?
No, I think. Hello, sir. We can with you, sir.
I saw a message saying participant cannot see my presentation. That's what I'm saying. If people are viewing, then that's okay. So we were saying basically that, we can leverage this knowledge to hunt, to become pathogen hunters, train them in order for them to get this pathogen, characterize them, and develop countermeasures. The world is in that state today simply because our ability to detect pathogens early, is not there. And it is when you have the ability to detect and characterize pathogen that you can develop the countermeasures among which the vaccine. This novel approach to science and then driven by genomics actually was during the 16 where we transformed outbreak response using pathogen genomics. So when this outbreak started in 2013 in the rural confinement of Guinea, nobody was was paying attention. Well, I gathered colleagues of mine at Rosemary University, and then the idea was to understand, I had a premonition that this was going to spill over, and this was going to spill over across West Africa. What were we going to do? So we identify, one of those things that was very important was diagnostics, because we knew if you cannot diagnose a disease, you can actually even manage it, or talk less of developing countermeasures. So we trained people across the region in Nigeria, Sierra Leone, and others, Liberia, and we were able to train them in order to actually use molecular diagnostics and also set up, leveraging the sequencing platform that we had across the region, studying other diseases in order to respond in case the disease was spilling over. As we rarely had a premonition, one of the scientists that we trained, Augustine Gober, did the first diagnosis of Ebola in Guinea, and we activated our contact tracing mechanism, and we're able to, in the first week, tested 35 suspected cases, confirmed 14, and also tracked down to the epicenter, who was a traditional healer. And that really actually started giving the confidence that we could actually do this. But what is most important here is the fact that, within the first 3 weeks of the pandemic or the outbreak or the epidemic in West Africa, we used next generation sequencing to generate the first 99 full-length genome of the Ebola virus. And we made it available, openly available to the international community for use to develop vaccines, drugs, and diagnostics. And in a real sense, we pioneered open data sharing and open data access. And that's what we're seeing today, with GISED, with Ebola, where people are depositing information into databases that have been used today to make vaccines and other countermeasures. So really happy that a movement that was started a few years ago has become the standard and then become the norm. And we did what we did in super quickly, at a speed that was unprecedented. That is 10 days actually from sample collection to sequencing and then depositing this in GenBank. And then our publication in Science could only come out about, we submitted a paper in Science 6 months later. So which means that we're really not out for fame, but we're really out to share and then making science openly accessible. But then we introduced some serious innovation into the way you could use genomics as a way to follow a pandemic. We were able to sequence across time, demonstrated that the epidemic was sustained by human-to-human transmission. There was no zoonotic introduction for animal reservoirs, and then there was no newly imported cases. In the case study of Sierra Leone, you could see this over time, and then you could see here where by June 16 we have one particular variant, SL3, that has taken over. This is the exact thing that we're seeing today with with COVID, where you can see once you have a new variant that is fit and then that can transmit better, like, you know, the Beta, the Delta, the Omicron, you know, that takes over. So we really demonstrated this for the first time during an epidemic, you know, in Sierra Leone in 2014, and also helped, you know, at the time, Nigeria, you know, save the day because we were able to use, you know, this technology and work with public health authorities to avert what could have been one of the major health disasters that is helping Nigeria to to contain that epidemic in 93 days, resulting in only 20 cases and then 8 deaths. That was unprecedented. But then in addition to that, what we did was actually to provide evidence of host-to-host transmission and then see how the virus was evolving, see accumulation of mutation across the genome. As you can see here, across the genome of the virus, we identify a few mutations, 27 of them, and we're able to demonstrate that those mutations were involved actually in driving, you know, increased transmissibility. And that was the first time where during an epidemic, we could use genomic evidence to show evidence of increased transmissibility, you know, of a virus. But with all of this information, we're able to put together, and you could see through this map, as you see here, within the space and time, we use genomic sequence data to actually show transmission from person to person, from county to county, district to district, country to countries. And that's really the first time you could show how you could use genomic actually to see how you could drive out the drivers of the epidemic and then use this as, for public health action. And that is really what you call translational research. But in addition to that, we realized that, you know, during the Ebola outbreak, you know, the turnaround time for detection using PCR or real-time PCR is very long. So we really went on and then translated, you know, the sequence data that we have, to develop what we call the first Ebola RDT, and which was the first generation Ebola rapid diagnostic test that was approved by the WHO and then the FDA under emergency use authorization. So this really, along with other measures, introduction of this rapid diagnostic test became a major turning point to curb this pandemic when it was introduced in Africa. Now, the second use case that I want to show here is actually to show how, you know, translational research can actually turn around a whole lot of things. We generated by 2015, we generated the largest Lassa virus genome sequences in the world. And bringing this data together, you know, for a disease that was diagnosed in Nigeria over 40 years ago or 50 years ago, and then there was no diagnosis locally, we not only set up the first local molecular diagnostic for Lassa fever, we conducted the largest, you know, the first genomic sequence of the virus, but then those sequences making them available actually changed the paradigm of a disease that was ignored. Between 2015 and today, when we actually generated the first sequences, there have been 3 vaccines that have been developed, and 3 of them, several vaccines that have been developed, and 3 of them are presently undergoing clinical trials in West Africa. We made these data available, but the bottom line here is actually the fact that we did not just only generated the data, we also developed, use the data to develop another rapid diagnostic test, which is the Pan-Lassa rapid diagnostic test you see here that has been deployed. But that also, the sequence data also made us, you know, have a better insight into the virus. The origin of this virus present, we estimate that it was actually about 1,060 years ago from present-day Nigeria, was circulating within Nigeria for 400 years and then moved out of this country for circulating in Nigeria for 600 years and moved out of Nigeria 400 years ago toward the West. But then what is important is the fact that we actually breached the gap and then drove, you know, innovation by making this available and working with the colleagues. We have 3 vaccines in clinical trial. So which means that the future is no longer bleak, you know, for this disease. More importantly is actually the fact that in Nigeria in 2016, in 2018, there was a public health emergency. Many people were dying of Lassa fever, but then we had to work with the Nigeria Center for Disease Control to answer these questions. We actually deploy again next generation sequencing technologies in collaboration with the Africa Center for Nigeria Center for Disease Control, sequenced over 200 full-length viruses, but then also demonstrated that, you know, the outbreak or the epidemic was due to, you know, there was no evidence of particular new virus strain, no evidence of increased sustained human-to-human transmission, but it was due to multiple introduction of genetically independent viruses similar from known lineages in Nigeria from animal reservoirs. What is important is also the fact that we demonstrate that based on depending on where you were in Nigeria, we had different lineages circulating. In the northern part of Nigeria, we were lineage 2. In the western part of Nigeria, it was lineage 2A, and then in the eastern part was lineage 2B. We had this published in New England Journal of Medicine, but we also demonstrated clusters of nosocomial transmission from patient to doctors, and that really was very important for making the policies in order to contain this epidemic. But we took this a little bit further, a step further, leveraging, the emergence of technologies like the CRISPR, which is gene editing technology, to develop a new, generation rapid diagnostic test for Lassa fever that is actually being deployed now. So you can see here, basically just using, that's just showing you using, technologies and machine learning, we're able to identify, regions that are very unique to the virus and develop guide RNAs around them, and then really use the appropriate nucleases to actually cut, and then that give us a result. I mean, with the reporters, then give us a result like this one. Double bar will show you a positive, and negative bar will do one. So we had an embedment, Amy Maxon from Nature was actually embedded in our lab for a week, because this was really the very first better and cheaper, CRISPR-based diagnosis for an infectious disease. And then we got this published in Nature, but this is showing, a student in my lab, and this really thanks to them because they did an incredible job to drive this and then this innovation and translating the genomic data and the genomic data into point-of-care diagnostics. The other thing that I want to point here is the fact that, yes, we do a lot of bioinformatics research in our lab, but we really translate them and then make them available. Here you can see here one of the pipelines that we developed for, viruses, for viral assembly, but we also develop another pipeline for bacterial genomics. Now let's fast forward to COVID, which is really the crux of the matter here. So when COVID started, in an unprecedented collaboration, a very successful collaboration between the Lagos State Ministry of Health, Nigeria and Nigerian Institute of Medical Research, the Nigeria Center for Disease Control, sample got down to a lab, and then we were able to sequence that on an unprecedented speed, 48 hours. This is a feat that is yet to be met by many centers or many labs across the world. So in 48 hours, we were able to establish the first genome sequence of SARS-CoV-2, not only in Nigeria, but in Africa. And then knowing fully well that, this was associated person, we were able to say clearly that this person got infected in Switzerland before bringing the sample, bringing the disease in Nigeria. So that really became really what I call the driver, that showed Africans that they could do this, and then that could also do this. That they could do this. We created this dashboard that we actually were giving, the Nigeria Center for Disease Control in real time, as you see here, the different lineages and variants that were circulating in Nigeria. This is just showing you an example. So, and then giving them real-time data that will make them intervene. Again, this is translational research, the way you want to view it. This gives you, like, over time, the different lineages that came to Nigeria. You can see here, this is in collaboration with many institutions across Nigeria, NIMR, and then many other institutions across the countries getting the samples. And then we really had this, and it will help, apart from Nigeria, we also help sequence sample from over 30 countries in Africa, and then train people along. And you can see here in collaboration with the Africa CDC, the map of what happened across the continent, and helpful. But in order to translate the genomic data, we also develop another rapid point of care test, and then called SHINE, and it is also based on the same principle using CRISPR gene editing technology in order to create. It's more like you are doing PCR on a sheet of paper. So this is at room temperature. You need no equipment. You need nothing, and then within minutes you can get your result. And that this is really revolutionizing the way we're really doing science. But then what is important is the fact that we've gone ahead and then really did something. This is showing the validation of the first 500 tests. We could do that within weeks, and this is within 8 weeks. But what is important is the fact that we've now been able to do a massive, multiplex of 169 different pathogens in one single test. And then this is really translational genomic research, because now we have the ability to diagnose and detect all of those viruses in one single test and then provide a result. Now, we also developed the workforce, so which means workforce development is very important for translational genomics. So we train people across Africa. You can see here, we've trained over 32 different African countries now. And then that has really increased dramatically, the output of genomic sequence in Africa. You can see between March 20 and 2021, there is a huge spike and an increase in the number of genomic sequences. So we made a milestone of 50,000 genomes way before the deadline that was given to us by African CDC. We've identified, B1525 variant, but while other countries in Africa have developed, the beta variant and others. But that's really just pointing out to the fact that now with translational genomics, we've been able to not only expand the scientific capability of Africa, we've also enabled Africa to start doing independent surveillance for outbreak response. Now, this takes me to that, to that questions. And then in addition to this, again, I just want to pass that to now narrow down to the vaccine. Did we attempt to develop a vaccine? Yes. What we did was basically to use, a platform that we're using before, developing a vaccine for Lassa fever, in Nigeria, that we just leverage it in order to develop a COVID vaccine. That goes from developing sequence data. And then from those sequence data, we use machine learning to identify antibody-dependent enhancers, take those domains out, and then you could actually increase the breadth of your vaccine. By that, you increase the breadth of your vaccine protection. So we did an in vitro screen from those synthetic gene production that we can use, and then plug that into the pseudovirus. So we did in vitro screens. And then from there, we selected a few candidates that underwent clinical trial. The question is, where are we with this clinical trial? I can tell you, proudly that this vaccine is undergoing clinical trial in the UK right now. Now, the question is, did we try to do that in Africa? Yes. But then why are we not doing the clinical trial here? Because it was, they didn't find, or African governments and countries did not find it interesting in supporting, a vaccine moving forward for clinical trial. And that led me to the next question. How close is Africa to vaccine production? I will tell you, not close at all. It will take a few years, minimum 5 to 10 years. And then the reason is simply because we don't have the infrastructure, we don't have the know-how. And Africa used to produce vaccine for some infectious diseases plaguing the continent in the 90s. But then in 2020, there was no single African vaccine manufacturing plant. So what happened? Neglect. Africa is supposed to be investing into the areas of vaccine production. But what political will, there's a need for investment by government and then the private sector. And I believe with that, we should be able to do it. But I want to leave you with some thought. And then the reason why Africa should invest in this area and should go into vaccine production. Africa urgently needs to guarantee its own security. That's the first thing I want to say here. This is a piece in Nature that I published, early this year with Dr. John Kingerson. The second thing is that Africa could build on the astonishing gain that it has made in surveillance and public health responsiveness, as I mentioned in recent years, to sufficiently invest in commodities to ensure its health security and position itself as a health leader in fighting infectious diseases. And ultimately, I believe that Africa dependency on the outside world sustain a lack of confidence in Africa, both within and outside the continent. Thirdly, I will say, and as a fourth point, as a SIP, and this is really the reason why we are where we are today, is simply because since African countries gained independence in the 1960s, the continent has largely depended on outside world for its health security commodities. We're talking about diagnostic, therapeutics, and vaccines, as well as personal protective PPEs and other medical supplies. So the COVID-19 pandemic has exposed how easily international cooperation and multilateral agreement can be dissolved, because other countries have a bigger problem, especially in the face of a global crisis. And this shows how vulnerable, this dependence leaves Africa. The fifth one, I will say, do we have an alternative as a continent? The answer is that we have none. And if we do not work toward guaranteing, self-sufficiency, we will fail to address the infectious disease threat of the 21st century. So it will thereby we'll be unable to achieve the developmental goal that is encapsulated in the Agenda 2063, which is the Africa we want. That's the blueprint by the African Union. But finally, I will say that multilateralism is always very critical in preventing and responding to epidemics and pandemics. But Africa will be able to benefit from this for the advances it has made in disease surveillance only if, its approach to public health is reconfigured towards self-reliance, among which vaccine production. Thank you. And I want to also acknowledge everybody that has funded our research over the years. Over.
Thank you very much, Prof. We have been able to listen to Professor Christian Happi, who talked extensively on genomics in Africa, cutting across different countries with much more emphasis on events in Nigeria, the Ebola, the Lassa fever virus, and of course to the current one, the SARS-CoV-2. Challenges have been mentioned and raised by Prof. I'm sure the participants have noted and jotted down a series of questions. Some of them have started even coming in, but without wasting our time, we want to go to the second speaker, and that is no other person than Professor Rosemary Audu. Thereafter, we'll take questions together, and we'll be glad to close the session afterwards. But without wasting time, let me call on Professor Rosemary Audu. Professor Rosemary Audu is the Director of Research, Nigerian Institute of Medical Research, that is NIMR, in Yaba, Lagos State, Nigeria. She's to present to us Nigeria Molecular Laboratory Network Beyond COVID-19. Prof, you are welcome on board. Over to you, ma. Thank you. Professor Audu, over to you.
Yeah, good afternoon.
Good afternoon. You're welcome.
Thank you. So let me share my screen straight away. Okay. I hope you can see my screen.
Yes, we can see, Thank you.
All right. Let me start by thanking the organizers of this webinar for the opportunity to speak here. And I will go straight to my presentation. Okay, video. So let's see. All right. So I'll be speaking on Nigerian Molecular Laboratory Network Beyond COVID-19. So let's look at what laboratory networks are. Laboratory networks are established to provide accurate and timely laboratory confirmation of infections. They are essential components of disease surveillance systems, and they provide high quality surveillance data to help guide disease eradication, elimination, and control programs. Having a network of laboratories will help support public health response. Now, due to the surge in several infectious diseases globally, effective tools for prompt diagnosis is required. There are several techniques for detection of pathogens, and they include serology, culturing, plaque reduction, neutralization, and several others. But these take time to get results. The turnaround time is a bit long. However, molecular technique has improved testing and reduced result turnaround time. Molecular labs, we have some challenges and solutions. Some of the challenges with molecular laboratories include non-availability of well-equipped labs, lack of trained personnel, and then cost of reagents. And the solutions are to equip the labs, well-equipped molecular labs setup and training of scientists, and encouraging in-country development of kits and reagents. WHO has prioritized some diseases for research and development. In 2017, they did this. And this means that these diseases are things we should begin to put surveillance on them and look out for them. And they include hemorrhagic fevers such as Lassa fever, Crimean Congo, and Rift Valley and Zika viruses, amongst others. Now, we're going to look at Nigeria's effort to, let me see, I'll move this. Let's move this. Okay, Nigeria's effort to, this is showing, where am I? Okay. Nigeria's effort to strengthen laboratory networks for improved diagnosis. So in 2016, since 2016, NCDC has been strengthening its own capacity for as a public health institute for response to outbreaks. And previously, efforts in networking was focused on single disease programs such as the polio. We used to have polio labs, HIV labs, and TB labs. But integration actually is the way forward. NCDC, the lab in NCDC, that's the National Reference Lab in Gadwa, has the diagnostic capacity for national priority diseases, including yellow fever, Lassa fever, monkeypox, cerebrospinal meningitis, and some other enteric pathogens. And it also has a specimen referral system that has been established, okay, where samples are sent to them for testing. There is also a national lab network that's of supporting 41 facilities with a network that conducts testing. In 2017, NCDC also responded to a widespread of outbreaks of meningitis, monkeypox, yellow fever, cholera, and even Lassa fever. We'll look at some of the efforts and concerns. Well, they have currently involved in advocacy, as an important tool for diagnosis of outbreaks. And some challenges that they have, that we are experiencing include the increasing disease outbreaks and emerging antimicrobial resistance. So building lab systems for disease surveillance and timely outbreak detection and containment is critical. It's very, very important that we have surveillance systems such that we can pick out these diseases even before it spreads. And if we look at the story of Ebola in Nigeria, that was just the saving grace for us. So to do this, we need efficient coordination, integration of surveillance and lab systems. Also, improving access to diagnostics at the country level is very key. Then improved patient management will be the outcome of this. Now let's look at challenges and successes in laboratory integration with 2 disease networks in Nigeria. Looking at the yellow fever, the Nigerian Yellow Fever Lab Network was established in 2006. It consists of 4 labs located in Lagos, Kaduna, Abuja, and Gombe. They have capacity to implement IgM detection using the yellow fever diagnostic algorithm. And at that time, they were receiving reagents for ELISA from Institute Pasteur and USCDC. And these reagents are in-house developed reagents. And then for them, at that time, they will always, and I think up till now, they will always send their samples to Institute Pasteur Dakar for confirmation. Now, what were the challenges that we have experienced with the yellow fever? Lack of reagents. Between 2016 and August 2017, there was no yellow fever testing in Nigeria because there was no supply of kits from Institute Pasteur and USCDC. As soon as the kits was received in August 2017, there was this report of the first outbreak in Kwara State. And then also, we have the challenge of delayed confirmation of a confirmation report because you have to send the samples to Dakar for confirmation. The index case was reported on the 16th of August, and then the confirmatory result came back a month afterwards, 12th September 2017. This does have implications for spread. And of course, also, even to get vaccines from the international body would become challenging because there has to be a confirmation of the outbreak before they will release vaccines. So this delay, it actually will cause a spread of the infection in the country. In 2018, ACEGID reported 9 cases using qPCR. They still had to send the samples to Institute Pasteur And the results came back on the 31st of December, 2018. So there was just this delay. They would not accept the results of the qPCR because molecular testing was not in the algorithm. But the solution now is that the National Yellow Fever Testing algorithm has been updated to include molecular testing, and the labs have been registered in a qPCR quality assurance program. Now, looking at Lassa fever. Previously, testing was performed in Sierra Leone, not in Nigeria. But between 2005 and 2012, the diagnostic capacity of 2 labs was established.
Sorry, Prof.
University. Hello, can you hear me?
Yeah, we can hear you, Prof, but I think your slides are frozen. We are still on slide 6.
Pardon?
We are still on slide 6, so I think it's frozen.
Oh, I see. But I'm moving the slides. I'm on slide 11 now. So maybe I'll stop sharing and start. I will resume. I'll stop sharing, then start again. I'll continue from there.
Yes. Thank you, Prof. Thank you. Yeah, thank you.
So, what slide are you seeing now?
It's fine now. It's fine, Prof.
Yeah.
Okay. All right. Thank you. Yeah. So, for the Lassa fever, I said earlier that previously, samples were sent to Sierra Leone for testing, but between 2005 and 2012, the capacity of Lagos University Teaching Hospital and in Irua Specialist Teaching Hospital was established. By 2018, we reported more cases of Lassa fever, and this was due to the increased awareness, improved surveillance, and lab capacity. Then that 2018 also, we had an increased number of collaboration from international community, to improve Lassa fever detection, treatment, and prevention. Through donor support, 2 more labs were established and included on the network. And these are the National Reference Lab at Gadwa and the Virology Lab of Federal Teaching Hospital in Ebonyi State. The key success factor for this Lassa fever, for this network, is that is the adoption of the testing algorithm based on 2-gene target testing strategy using the qPCR, with a combination of both commercial and lab-developed tests. Then we look at the COVID-19 era. As at January 2020, we had only 6 biosafety level 3 labs with capacity for SARS-CoV-2 testing. These labs were testing for Lassa fever, yellow fever, and cholera using qPCR. And then these labs soon became overwhelmed with increasing COVID-19 incidents in Nigeria. By April 2020, Nigeria could only test 2,500 samples per day. So NCDC leveraged on 2 existing platforms, the HIV molecular lab platform and the TB Gene Expert platform, and they repurposed these instruments for SARS-CoV-2 testing. So as at 2021 April, we now have 124 COVID testing centers in the country. 76 of these are public owned and they are within the NCDC network, while 41 are fee-paying private labs, while 7 belong to corporate labs. Now we're going to see the NIMR contribution to molecular diagnostics. And most of the things here in NIMR are homegrown. They are things that we have used majorly funds from within the country to do, not much of grants. So NIMR's role in molecular diagnostics dates back to the 1990s, where we have used molecular technology for diverse research. In 2001, we now commenced molecular diagnostic services. And currently, we have an array of assays that we do in NIMR. The HIV-1 viral load, hepatitis B, hepatitis C, viral loads, HCV genotyping, HIV DNA PCR, HIV-1 resistance testing, HPV screening and genotyping. And we also screen for viral hemorrhagic fever, 7 different viruses. We do this routinely. Then we also in NIMR have the international recognitions for molecular diagnostics. In NIMR, the Center for Human Virology and Genomics was the first ISO 9001 certified lab in the country, diagnostic lab. And in 2017, we also obtained the ISO 15189 accreditation. By 2018, we're listed as a WHO pre-qualification lab. In this case, it means that assays that will be used at the global level have to be pre-qualified by WHO, just like before we use kits in Nigeria, NAFDAC has to approve. The same way at the global level, WHO has to pre-qualify. So WHO has 13 labs globally that can evaluate these kits for use. And NIMR happens to be one, and it's the only one in the West Africa. And the assays that we are listed for include HIV, hepatitis B surface, hepatitis B and HCV, both serology and the nucleic acid test. And then also in 2021, NIMR lab was designated a WHO National HIV Drug Resistance Lab. Now we are going to look at some local kit production for COVID-19 and other viral pathogens. Like I've said, all we have done here, and I'm reporting they are homegrown. So in NIMR, we have produced DNA and RNA extraction kits. These are readily available at low cost, and they can extract nucleic acids from various specimens. In fact, that is what we use right now in NIMR for most of our studies. Then we also produce the isothermal PCR point of care. This is useful for decentralized testing phase. We also produce the production, the qPCR assays for SARS-CoV-2. We call it SCODA in short. This kit is in-house kit, and it is the kits that, this kit we have actually, it has been commercialized. It's a kit that we're using right now in NIMR for testing from our drive-through. And then also just to mention that the Roach kit that we were receiving from NCDC, I think we had a stock out since either August or September 2021. And because of the global demand, they said the next supply will be in February. So between that time, we're just using our own in-house kit that has been validated for testing testing COVID-19 for free. We also have developed the sample release reagent. This one is, the advantage is that it's less hands-on, complicated lab setup and highly skilled staff is unnecessary. And some of the productions that are ongoing right now, we are still conducting research on this, are the vaccine production using the CRISPR transfection technology. And also we are working currently on producing ELISA test kits for COVID-19. Some additional capacity that we have in NIMR. We have produced other test kits like the Lassa fever and the yellow fever qPCR test kits were produced in NIMR, and these were launched. The Minister of Health was in NIMR and they were launched. And right now, as we speak, we are just at the phase of going towards NAFDAC registration. Then we also have some other capacity in NIMR. We have had the standard Spencer since 2008. In fact, just like Professor Happi mentioned, we jointly sequenced the index case, even though ours was a partial sequence. We also have the next generation sequencing. Currently, we have sequenced over 200 samples using that platform. We also have the third generation sequencing platform, the mini ion. We are currently sequencing over 300 samples, and the Omicron variant is dominating. That is going on as we speak. We have a well-equipped toxicology lab with gas and atomic spectrophotometers. We also have oligosynthesizer that was provided by MTN, and it will help in the production of primers locally. And these things become quite important because at the outbreak of this COVID-19, reagents became really scarce because of the global demand. So if we are able to produce them in country, I assure you that it will go a long way to help our research and diagnosis. Now, I just want to quickly speak to a project we have here in NIMR that is going on. It has to do with trying to control viral hemorrhagic fever, the rodents, and the reservoirs, we're looking for them. And this project, we found that viral hemorrhagic fever, they are associated with one or more species of either rodent, bat, tick, or mosquito host. And then determining their host enables distance, and it will also help to design effective control measures if we're able to identify where the hosts are. And then Nigeria has reported several outbreaks of viral hemorrhagic fevers. So that's very important host reservoirs in order to interrupt the cycle of transmission. So we have these grants that we have awarded as extramural grants. And we're working in collaboration with some universities in Nigeria, and we were able to select some postgraduate students. In the last cycle, the cycle we had in 2019-2020, we had 5 students, 3 of them MSc students and 2 PhD students. They were looking at different reservoirs. We had some working in Ondo State, looking for yellow fever in mosquitoes, Lassa fever in rodents in Edo State, and dengue, yellow fever, Rift Valley fever in mosquitoes in Osun State. And we have the last person there working on Crimean Congo hemorrhagic fever in ticks in Kwara State. The last person has completed his PhD. This other lady is still continuing with her PhD. These 3 have completed their masters and they have moved on to their PhD program. Currently, we have 6 students that are with us on this program, and we are supporting them because we believe strongly that in identifying where these reservoirs are, we'll be able to break the chain of transmission. So looking at the post-COVID-19 era, we believe strongly that laboratory networking is very necessary. And then training on best practices for molecular analysis is essential. Attaining international standard in laboratory operations is important because if we don't have that standard, whatever results we are generating will not be accepted internationally. And then collaborative research indeed is the way to go. So for those who are interested in setting up laboratory network, it is a duty of the public health institution like NCDC. These are the steps that are required to identify a team that will lead the development of a network, define the purpose for this network, develop a broad proposal, plot the cost, identify potential needs, make contact with potential partners, and then select and conduct lab network development activities. They are very essential, and I think it's the way to go. So I want to thank you for listening. Thank you.
Oh, thank you very much, Prof. We really appreciate your kind presentation. And without wasting time, we need to proceed to Q&A. So first, I would like to start with Professor Christian Happi. You know, he has been able to educate us so much, talking about the RDT, kit that was produced in ACEGID. Prof, how far are we with the marketability of the RDT kits? Can we have more of the kits in the market? Can ordinary laboratory, either private or a public lab or from the other sectors, can they go to the market and pick any of these kits? That's one. Then two, you raise the challenge of the clinical trial in UK or Europe and the problem with the African leaders. So can we go to the level of the African Union to raise such challenge so that we can start thinking of having clinical trial in any of the willing countries in Africa? And one more for you, Prof. The health security challenge in Africa. What are the recommendations you give to us, to resolve that? Thank you. So maybe we take from Professor Christian Happi first before we go to Professor Rosemary Audu. Thank you. Prof, over to you.
I can start with your last question that has to do with the health security challenge. So I think investment in research and development and then investment in supporting investors to produce consumables into this space is very important. The Abuja Declaration requires that African countries spend 15% of their GDP on health. Very few countries are doing that. 2% of the country GDP should go to research and development on health. No country in Africa has met that. Maybe Rwanda is very close to. So those are investment and education. That's what is needed. To your third question, in your health security. So that's the point. Now, related to, I think your second question had to do with, if I'm not mistaken, had to do with
the clinical trial.
The clinical trials.
Yes, and the challenges.
The challenge is actually funding. So if you have a vaccine and then you don't have clinical trials are expensive. If you don't have the resources, then you can't do the clinical trial. So we had a COVID vaccine developed, but no country, picked it up in Africa. So eventually the UK, that vaccine is undergoing clinical trial now in the UK because they saw value because it was a broad range, corona vaccine, and that definitely they saw value into that, and then they funded it, and then that is going on there. So it is about funding, and it's also about getting the resources and then the infrastructure. Very few countries in Africa do have infrastructure for vaccine clinical trial, even though there are some experts around, but then with resources, you can actually beef up the existing infrastructure and then have clinical trials and build the capacity and then have it done. And that's the reason why I said, when we talk about vaccine production and then having all of it, we still have a long way to go because there is a need to actually invest in this space. And that is important. Investment, again, we're getting back to the political will. Are we willing to do this in order to improve vaccine production in Africa? That's the question mark. So the African Union should be able to take that. So you raise the issue about the African Union taking responsibility. Yeah, maybe now we're having some countries, making moves toward becoming vaccine producers, but it's going to take time. We have Rwanda, we have South Africa, we have Senegal, we have Nigeria that is coming up, and Ghana, and then maybe Morocco. But it's going to take time for us to get to a level where we can produce because there's really no infrastructure. It's going to take time to build those infrastructure. It's going to take time to build the capacity, human resources in order to do that. Finally, related to the diagnostics, yes, we did produce the diagnostics, and then the diagnostics, we're using that a whole lot in our lab, and then we validated that in many setups where we're working. I just want to say that that diagnostic has received US FDA approval, and then is actually being used, in the US. We definitely, the only reason why we are not having it, in the market right now is actually the fact that, raw materials becomes a problem. We're having them in trickles. So, and because of many countries in the West, and again, it gets down to the point whereby we cannot even produce raw materials. Many countries in the West really have put embargo on a lot of the raw materials that we're actually producing. You can imagine I ordered a few strips from Germany, sometime last year in order to produce, to spread this test around. It took them over 8 months for us to get strips down here. So it's a big challenge, and it is the fact that we don't have the industries or the local industry that can produce, the necessary consumables and raw materials that we can use to actually democratize and make this test available. Simply because we basically have moved the technology beyond, looking at antigens or antibodies. We're basically using gene editing and then having the using CRISPR, producing all of the RPAs and other things that are actually needed in order to do these tests. We don't have facility to produce them at the moment. Over.
Okay, thank you, Prof. We'll be taking questions from the participants. You can please indicate you can test, by writing in the chat box. And likewise, you can raise your hand if you intend to speak. So, but before we do that, and likewise, we also have members of the panelists that have been interested in raising 1 or 2 questions. But before we go to that, let me just quickly raise 1 or 2 questions to Professor Rosemary Audu. Prof, the first question to you, ma, is that you mentioned the issue of the yellow fever, testing in the country since 2006, and that we have a lot of information in country. So what are we doing with vaccine production that was stopped over time? How do we resuscitate that in the country? Secondly, you talked about the algorithm, challenge about the qPCR that was detected, that was used in ACEGID to even detect the yellow fever virus. And unfortunately, you couldn't incorporate that into the algorithm, until lately. So what are we doing to prevent, such a reoccurrence, of that? And lastly, for me, Prof, is that you talked about the laboratory network, the coordination. How do you want the coordination to be done? How best can we handle the coordination, for things to be, rightly perfected? Thank you. Over to you, Professor Rosemary Audu.
Sorry. Okay. Yeah. So thank you very much for the questions. Yellow fever tests and vaccine production. Honestly, ironically, yellow fever was first detected here in Nigeria, specifically in the Nigerian Institute of Medical Research here several years ago. And we had the yellow fever production lab here in Yaba in Nigeria. But for some reasons, it ran down and they have not been able to sustain it. But I believe now, I think it also has to do with maybe political will, probably do not understand the importance and are not ready to fund it. And we probably haven't gotten, we're always looking for donors, always looking for people to support us. We haven't gotten anything, probably, that's why. But it was working and functioning. I was in Dakar. That of the production lab, the yellow fever production lab is functioning in Dakar. But it's unfortunate. But right now, I know that we have attended 1 or 2 meetings where they are thinking of revitalizing these vaccine production labs. There's a lot of politics to it. I think if we remove politics, there are many things we can do in country. If we remove politics and we are ready to fund research, I'm sure that we'll be able to accomplish a lot. Just like this COVID-19 outbreak, we had this intervention funds that came in. And with that, we're able to do so much that we didn't know we could do. All right. So if within us, within the country, we support this effort, I'm sure it is something that is doable. It is doable. Then for the yellow fever algorithm, I mentioned that as at the time they were still using serology for testing, ACEGID had used qPCR to report 9 cases. But because the PCR was not within the algorithm, because it was not within the algorithm. They had to send the samples again to Institute Pasteur in Dakar. But right now, I mentioned in my presentation that it has now, qPCR has now, the algorithm has been upgraded to include qPCR, and the labs have also been enrolled for quality assurance. Okay, so I believe now that PCR is the way to go for yellow fever diagnosis. Lab network, how best? I want to say that NCDC has done so well, especially with the COVID-19, starting from 6 labs to 124 as we speak. I believe if they use a similar approach, they can expand the scope, you know, because they just have scope for each lab has its own scope. It's not the fact that you're in a network does not mean that you can do all the diseases, no. But if they open it up, there are many more labs that have capacity to test for some of these outbreaks and notifiable diseases. Thank you.
Okay, thank you, ma'am. So can we take questions from the chat box? I can see people raising up their hands. So question to Professor Mr. Happi, what are the opportunities for training faculty members in your institute? And that is from Folusho Ologundudu.
The opportunities for training faculty members are existing. I think in the past 2 years, we haven't trained, but prior to that, we used to have several training opportunities. We have short-term trainings in courses like genomic sequencing, short course training in bioinformatics. We have a summer, we'll call it summer genomic bootcamp. That is an international training program that brings people all across the world. So it's the opportunities are many. I think for the person is actually to reach out and then see, and then it's all depend on exactly what we want. I also want to point out that we don't provide all. The training is not indiscriminate. So people just bring to us requests that we cannot fulfill. So I think it has to be studied and examined and see whether we can do it.
Okay, then Prof, do they have to come in person to the university, or can they get to the website and download form or something?
They can go to the website, and then there are forms, there are calls, and then they can always reach out to info@acegid.org. And that, there are people that are dedicated to respond to your questions.
Okay, thank you. Second question from Zainab Aliyu. This goes to Professor Happi again. You talked about mutation found out after sequencing of the genome of Ebola virus, and it's been responsible for spread of infection. Please, how long does it take for mutation to be detected in the virus, and what are the other causes of mutations?
Well, we need to understand that, especially RNA viruses are very prone to mutation. We understand that. But then the mutation that are acquired by, remember that each mutation, when the mutations are acquired by the virus and they become fixated, it comes at a fitnet cost. And then once a virus has acquired a mutation, especially non-synonymous mutations, you will see that, they are often acquired for the purpose of a beneficiary purpose. And in the case of Ebola, for instance, we were monitoring the virus and it spread, over time. And it was about 5 months after we had the first cases that we started seeing this mutation, and then they started accumulating. And then having discovered that, then you can relate that to, increase in number of cases, but we also have to do functional analysis and then do in vitro experiment to relate that to increase transmissibility, just the same way it's done for SARS-CoV-2. What I mentioned then, that it was just the first time we actually were using this approach in order to relate that, to transmission and disease spread.
Okay, thank you, Prof. So, Professor Rosemary Audu, is there any current collaboration with the veterinary research laboratories? And you can take that with this, ma. Is there for surveillance individuals and lab, and that's a database where people can actually, thank you.
Sorry, I didn't get the second question.
Okay, is there a central database for surveillance carried out by individuals and the laboratories?
Okay, thank you very much. So whether we have any collaboration with any veterinary lab,
Research
yes.
laboratory, exactly.
Yes, yes, yes. So we are working in collaboration with University of Ibadan, University of Ilorin, their veterinary departments. So, I mean, for our viral hemorrhagic fever project, yes, we are. And then we are also working with VOM, working with VOM on the vaccine production. So we do have collaboration with vet laboratories. And then concerning the central database, well, database that I am aware of right now is what NCDC is monitoring, such that all the testing we're doing, we are uploading into the NCDC SOMAS website. We do that each day. We upload our data into the NCDC database. I hope that helps.
Okay. Hello, Prof.
Yes,
Hello,
can
Prof.
you hear me?
Yes, you can take that part again, ma. I think the network.
Oh, sorry about that. I said the database that I'm aware of is the SOMAS that the NCDC manages. So all the testings that we do, particularly for COVID-19, is uploaded daily onto that database.
Okay, I think the participants would want to push through you maybe to the NCDC or to the federal.
My network is bad.
Something that can we have such database, thank you, ma. So you can just help us convey that. So that's one. Then we have another question here that how readily available are the rapid and molecular kits, that you talked about for both Professor Happi and Professor Audu?
Okay, yeah. I'm no longer a panelist.
They are developing house in NIMR. So how readily available are they? And the same goes for Professor Christian Happi, who talked about the Lassa fever, the Ebola RDT kit. Thank you. Over to you, Prof.
All right, let me start from NIMR. The kits that what we have developed in NIMR, the RNA extraction and DNA extraction kits are available. So it's just available for the asking, and it's quite cheap. I think, how much is that? It's about 50,000 Naira.
Okay, so we need
tests.
to come to, so can we come to NIMR to pick that up?
Yes, you can. You just place your order and you will receive it.
Oh, thank you.
And then we also have the Skoda kits for testing of COVID-19, but we said that has been commercialized. So we can give you the name of the distributor and you can access it.
You said that as the what, ma? You said that as
the what, ma? Commercialized. Okay, we have sold that out, and the distributor can always, you can always place your order with the distributor.
Okay, then for the primer, you talked about the oligo sequencing within. So how do we go about that? Can we just send the primer sequence and you forward?
Yes, okay. Exactly. So from our website, you can always get that, place your order.
Okay, thank you, ma. So let's take from Professor Happi. Prof, you want to respond to the same question?
Yeah, the Lassa fever tests are already available. So I think those are antigen capture tests. So they are all available. So if you want to look there, I think they are commercialized, they are commercial partners in Nigeria that supply them. And for the COVID test, as I said, we do have that in-house. We have not really commercialized it because of supply chain. We do the little that we can do in-house, but we have not at this level where we scaled up production to a point of commercialization.
Okay, well, thank you very much, Prof. So let's take from, we have more questions from the participants now. Can you please indicate so that you can be called upon to ask a question. Then the members of the panelists. Okay, while we are waiting for that, I think I can take more questions from the chat box. We have from, this is coming from Dr. Anima Shang, that the death rate in Nigeria and other African countries has increased, not because these people are infected with COVID-19 viruses, but majority died because of weak immune system. How can we understand the immune system through molecular biology and genomics? Professor Happi, you want to take that, sir?
Well, I don't understand the question, but I think if you say that the death related to COVID is due to weak immune system, we've seen it is.
Okay, the question is saying, how can we understand the immune system, using the molecular biology and genomic tools?
Well, but there are several ways to do that. I mean, there is what we call immunogenomics. You can look at, also you can do functional analysis system by studying, the immune responses. It could be either cellular or humoral immune response using, and then also look at cytokines that are involved into, immune response. So, and basically using, different approaches. If you wanted to, look at either T cells or look at B cells, there are different ways to do that. So I think, generally, at least what is a way that you want to do to see exactly if you want to present to certain specific epitopes, how the immune system respond. But I do believe that, what we're seeing for COVID is when you talk about people that are immunocompromised, and that's the reason why we say that people should get vaccinated. Those people should get vaccinated because when they get infected with variant like Omicron, then their system, they become overwhelmed and then they cannot control the infection. So that's the more reason to get vaccinated, and then the more reason why, people that are immunocompromised should get vaccinated. Studies have proven across the world that, those that are dying mostly now, not only in Europe and even within Africa, are people that are the unvaccinated people because their immune system is a little bit, is more overwhelmed compared to those that have received at least their doses of vaccination. Over.
Okay, then Prof, do we have the technology, the immunodiagnosis? Can people come around to your institute to collaborate or to perform assay on that?
Yes, we do have a spot assay in my lab. We have, and then we just established neutralizing antibodies assay also, and then that can be done. Over.
Oh, wonderful. Then, Professor Audu, there's a question here that what will be your view and expectation if we apply the laboratory networks, the technology that you've mentioned, that you talked about from NIMR, the development, of different kits and all that, to the challenge of malaria, and effective malaria vaccine development?
Yeah, so I think networking is still very good. Network is good for research. Collaborative research is good. So I think if we apply the same method, it will help advance this malaria research, because it looks like most times people are working in silos. You probably just on your own, you to yourself alone, and not wanting to collaborate with others. It will not help us. The best we can is that working with partners in the international developed world, they just keep taking samples away from here. We have come to find out that samples are a huge, huge resource. That it's not just for us to be shipping samples out, but in-house, we have those who are doing it abroad. Most times you discover that they are Nigerians. A good number of Nigerians are there. So we have the science. I think it's just the funds, and if we work together, we will be able to achieve much more. So if we use that same approach for malaria research, I'm sure we'll be able to have some breakthrough. Thank you.
Okay, then another question, Prof. A participant is asking that, do we have a national timeline for vaccine production? I think that should go to Professor Happi. Prof has mentioned the fact that we need up to 5 to 10 years, before we can get to the level of producing vaccine in Africa. But can we do something? We have a timeline, for that, so that we know the 10 years or the 5 years is 5 years. Thank you. Over to you, Prof. Professor Happi.
That is a policy question. I'm not the one to say the timeline for Nigeria. I think that question should go to the Ministry of Health.
Okay, thank you, Prof. But I'm sure you can, pass that over, in any of your meetings with them, that we need a timeline so that the issue of the timing, is crucial, and we just need to get to that level of vaccine production. Thank you, Prof. Then we have another question from Eboolua, saying that, please, are there any laboratory internship position in NIMR? That is for Professor Audu. Laboratory internship position in NIMR for a recent pharmacist graduate interested in molecular diagnostics and vaccine production. Over to you, Prof.
Yes, we do have interns, lab interns coming to our facilities. So it's quite simple. You apply that you want to do an intern, and if the department or the lab you want is, if it's the space, they will get back to you. We just have, I think, just a token, a small token that you will pay as lab fee or so, and then you can spend 3 months, 6 months, depending on how long you want to stay. And so there's provision for that. Yes.
Okay, Prof. But Prof, I'm sure in future Nigerians will be looking up to you giving them a waiver over the small tokens so that they don't pay anything, they just come around for the training. So I'm sure you will discuss that in any of your management or board meeting. Thank you, ma'am.
Besides that, we also have the NIMR Research Foundation, whereby on our website you will see there are adverts there for postgraduates and other researchers, they are posted there where we will sponsor them, we will fund them. Yes, even postdoc, yes, we have that. So if you go to our website now, in fact, there.
Okay, thank you, ma. Thank you, ma. Then we have another question from Dr. Williams Nabofa from Babcock University. The question is on COVID-19 vaccines. I'm sure that should go to Professor Happi. That looking at the way the virus behaved in Nigeria, do we have data to suggest how frequent vaccine booster dose should be administered? Professor Happi, please.
I don't think that we have the data. I mean, we are still struggling to get enough people vaccinated. And I mean, to say, I'm not sure that a study of that nature has been conducted to know how often vaccine boosters should be administered. I'm not sure.
Okay. But I'm sure, Prof, you'll be willing to, maybe your lab and other labs, to do something on that so that in future, the data can start coming out. Another question to you, sir.
Let me just quickly add that there is a network of labs that are working on that. It just started, it's very preliminary. Fractional dosing of COVID-19, and NIMR is in that network.
Great.
So hopefully we should have some data. But just like Professor Happi has said, we still have not, was it the last I heard was only 3% of the population have gotten those who need to be vaccinated have gotten the vaccines. So I don't know, we haven't gotten to that level yet.
Okay, ma. Another question, how long does the immunity last following COVID-19 vaccination in comparison with natural immunity? Professor Happi? How long does immunity last following COVID-19 vaccination in comparison with natural immunity?
I don't know. So I think, again, what is natural immunity? And then when you have natural immunity, can you define when exactly you started having that immunity? So there are a lot of unanswered questions. vaccines, again, when you talk about immunity to a vaccine, it is very dependent on exactly whether you are dealing with the same variant or not. If you have a variant and then you have a vaccine and it's not changing, I'm sure that immunity can be very long. I didn't think that, when people were developing COVID-19 vaccine, they anticipated all these variants that were coming, and then they were escaping both immune escape or vaccine escape. So I think if you're dealing with a disease that is not that, that doesn't change it that much, just like COVID or HIV, it's easy to say that, well, you can set up something that you have immunity for a long-term immunity. But in this case, it's very challenging. It's very difficult. It's very difficult for anybody to say today how long the immunity is going to last, because you don't know what is going to happen in the next few months.
Okay, thank you very much, Prof. I think
may I add?
Okay, I even want to call on you so that you can, go ahead, the chairman of the webinar committee. Yeah.
Okay, thank you very much. On the issue of the natural immunity, yesterday there was a report of a singer in Czech Republic who actually waited for natural immunity to kick in. She deliberately refused to get the vaccine and presented herself to get infected so that she can get access. So in that country, they have a policy that if you get natural immunity, is as good as the vaccine, so they can allow you to get access to crowded places. Unfortunately, she died on Sunday. So she got the vaccine, the virus. She was diagnosed positive, and she died because of that. So I think that it's still not a substitute for the vaccine. Natural infection is still not a substitute for the vaccine because of what Prof said there. You don't know what will happen when you say that you actually acquire natural immunity. So I think that actually answered some of our questions, that the vaccine is well controlled, natural infection not that controlled.
Okay. Thank you very much. I think we have those 2 minutes left before I call on the chair to appreciate everyone. I would like to thank Professor Happi, for giving us so much information regarding how far we've gone with the COVID-19 vaccine production in Africa, and of course the lessons from the genomics, the effort on the Ebola virus, Lassa fever, and several other viral hemorrhagic fever viruses. And likewise, to also thank Professor Rosemary Audu of NIMR, who has been able to tell us so much about the need for the laboratory network here in country, and what are expected after the COVID-19. I want to thank you and thank everyone. So over to you, webinar chairman, to give us your vote of thanks.
Yes, thank you very much, Dr. Anjorin, for a wonderful job. I believe, if not for time, we would have spent more than 3 hours having this nice discussion. It was an extremely powerful presentation. On behalf of the NYA, I would first of all like to greatly thank our speakers for today, Professor Happi and Professor Rosemary Audu, for a wonderful, wonderful presentation. You all will agree with me, all of us were much better in understanding these aspects of the pandemic than we were before the webinar. I would also like to thank the Nigerian Young Academy under the leadership of Dr. Mohammed Awal Ibrahim for providing the platform for experts to share their knowledge to a wider audience, whom comprise mainly of rising scientists and even medical practitioners that will actually be in the forefront in the fight against the pandemic. I would also like to thank the participants of today, very, very vibrant participants we have today. I can still see about 17 questions that are unanswered. So I will assure you that if we can get our speakers to answer your question, we will directly see that we share with you the answers to your questions. But meanwhile, we also would like to appreciate your participation, which we hope that the webinar is going to be a monthly event. We invite you to join us in our subsequent webinars. Thank you very much to the participants. Finally, we have to appreciate our sponsors under the COVID-19 Africa Rapid Grant Fund, which actually supported the event of today and whom are going to support our subsequent events that are coming throughout the year. Thank you all until we meet in our next webinar, which is coming up next month. in line with the economic hardship we are facing. We are going to have experts in the agricultural sector and the economy to discuss with us about how Nigeria can prepare to cushion the effect, the economic difficulties we are facing as a country. Thank you very much, and see you next time.
Thank you.
Thank you.
Chair. The individuals whose questions were not answered can send to our social media platform. We will get them across to the speakers with their email, and I'm sure they will provide answers to their questions. Thank you all. Thank you.
Thank you. Bye-bye.
Thank you, Prof.
Yeah, thank you so much. We are grateful to all the attendees and the panelists, to Professor Happi. Thank you so much, Professor Rosemary, Audu. We are really grateful. To all the attendees, we are indeed very grateful. And we hope to see you next month. Thank you.
Please, those who ask questions, can you drop your email, please? Drop your email, we'll get them, we'll forward to Prof, and they will answer it. They'll copy. Please drop your email.
Chairman, thank you. My president, thank you.
Dr. Anjorin.
Members of NYA, thank you all. Fellows and members, thank you all.
Oh.